what I understand the goals are tonight is to tell you a little bit about my career path and a few things that I learned along the way that might help you all. And then the second part is talking about the work that we do at the Santa Barbara Botanic Garden in the Conservation Department. So just a few things here at the beginning. So the biophilia hypothesis suggests that humans have an innate tendency to seek connections with nature. And these are a couple of pictures from my childhood. And mom was always saying, go outside, it's beautiful out. And we did. So we spent, my friend and I spent a lot of hours in vacant lots and drainage ditches, collecting frogs and whatnot. And just enjoying nature, as well as lots of outings and camping. I know that not everybody has had these opportunities, and I think we can all help to cultivate that in others by suggesting a hike, going camping, showing a nature show or a video, going to a garden, whatever feels safe for people. And I really feel that education and outreach at the garden is just as important as the work that the science work that we do. First stop on my career path, oddly, was photography major. And so the lesson there is nurture all parts of you. Few people know what they want to do exactly when they're 18. I liked the idea of being a photographer, although I don't think I really thought through what it meant to, as a job until I was close to graduating. So I went from wanting to be a fashion photographer to realizing I would hate the big city. And then I added a geography minor and thought maybe I could work for National Geographic or something. But then I realized I would hate the constant travel and the competitive nature of it. So all that said, the art skills that I learned and the different way of seeing will always be a part of me and embedded in the work that I do all along. So it all comes back to the education and outreach. As I look back through photos of my young adulthood, I see that nature was a recurring theme for me. My free time was often spent camping or hiking with friends and family. This is a picture of my sister and I. We backpacked to the top of Cone Peak in Monterey County. And I guess the lesson here is just listen to the signs. This is what I was drawn to sort of all along. It helps that the love of my life loves nature too. So I met John, and this was our first backpacking trip to Catalina Island. During that trip, he said, we should live here. He's the dreamer in the family. And I scoffed and said, where would we work? But little did I know we would come back there. We both went to school around the same time, with me pursuing a master's degree in biogeography at UCLA. And I didn't get in at first. So the lesson here is persist. I had good scores and I had good grades, but my application wasn't specific enough with research ideas. And a mentor of mine at Cal Poly advised me to find someone I was interested in working with and sell them on the research ideas that I wanted to do. So I did, and I got in biogeography. So this next one, this is my favorite quote of all time. The real voyage of discovery consists not in seeking new landscapes, but in having new eyes. And in that program, as I learned more about plants and animals, I really began to see more and more patterns, including, unfortunately, the invasive plants that are everywhere. But it opened up a whole new world to me. And I did a short stint in environmental consulting, and the lesson there is be true to yourself. While it's really good that we have environmental regulations, and it's really important that good people do that work, and the pay is better than most environmental careers, it didn't sit well with me to survey these areas that I knew were being slated for development. So even though it meant I would not be as comfortable financially as otherwise I might be, I went down the nonprofit path. And it was actually my husband saw the advertisement for the Catalina Conservancy, and I got the job, and it was quite an adventure. My work took me all over the island, studying and monitoring the vegetation and looking at the impacts of fire and invasive species. And I got a lot of great experience in different aspects of conservation, which has served me really well in my roles since. So I guess the lesson there is that first jobs will really set the stage. It helps you then get the next job. And so choose wisely in that first job when you have a little more, I mean, I'm sure there's different perspectives on this, but a little more flexibility as far as what you take, because it will influence your next jobs. So I did a whole lot of monitoring the vegetation across the islands to study different fires and the impacts of introduced animals like deer. Also general vegetation changes after the removal of introduced goats and pigs. I did a lot of mapping, from weeds to rare plants to creating a new vegetation map for the islands. I built a ton of feral animal exposures around the island for research and for rare plant protection. And this image on the lower right is a crew that we built exposures in a fire scar near the airport. And I did lots of exploring and botanizing. So it was just really good entry into the ecology world. But always photographing, of course, which I guess the lesson there is find your own unique niche. That art piece has always stayed with me. And I think it's really important to for the education and outreach. And I also set up photo monitoring projects that are being used to document changes today. As I went about my work on Catalina, I began to wish that I had more of a biological foundation that I had missed as an art major. I realized I was studying super rare plants like the Catalina mahogany in the upper picture on the right, and I didn't even know who pollinates it. And I was watching pollinators like this little bee fly in the upper left, and I really wanted to know more. So I entered a PhD program at UCSB with Carla D'Antonio and Tom Dudley. And I will admit this was a really, really hard thing. First of all, I don't recommend waiting as long as I did to go back to school. A lot had changed, and I was also entering a very foreign world of the hard sciences. Plus, I had a kid, a house, a husband, and a commute. And I found myself studying flashcards of plant physiology while on the slide with my kid. So I don't recommend that, but it was all about persistence, including the first year thinking I was studying fennel on Santa Cruz Island, and that didn't work out, and then starting a whole new project in the dunes. Opportunities struck early before graduating, 2 years before graduating, to get a job at the Botanic Garden. And thanks to my old boss from Catalina, Peter Schuyler, who told me about the opportunity, and the geography was just too good. So I got the job and I became director, and it came full circle to my first blurry photos of plants in the 4th grade on a field trip to the Santa Barbara Botanic Garden. So now on to what we do at the Botanic Garden in the Conservation and Research Department, where our organization's mission is to conserve California's native plants and habitats for the health and well-being of people and the planet. So we have sort of 4 layers of goals in our department, and I like to think of it as layers of a cake. You have to have the first layer. You have to know what the plants are. You have to understand the nature and the evolution of California's plant diversity before anything else makes sense. Which ones are rare? Which communities do they form? And then when we know which ones are rare, especially those that are rare because of us, we work to protect and recover our region's rarest plants, particularly on the Central Coast and the Channel Islands, but also more and more even beyond. And then the piece that I brought almost 10 years ago to the garden is the community ecology piece, to restore Central Coast habitats and food webs. And I didn't mention that I studied plant-insect interactions at UCSB, and so I brought that to the garden. But the icing on the cake all around is always that advocate, just, expose people to these things and the importance of conservation. So we do pretty comprehensive conservation work in our department, and I'm proud of that we do work from genes to ecosystems, because conservation challenges can be anywhere along that continuum. And this is our ever-growing staff. I ran out of room to even put their names. We're now a department of about to be 22. And we have sort of focuses of the botany and conservation genetics, and we now have a lichenologist. We have a growing rare plant conservation program, and we have habitat restoration as well as invertebrate ecology, which has the same goals, that we learn about those interactions so we can restore habitat effectively. So I'm just going to give you some examples of each of those goals. And the first one, of course, is understanding California's biodiversity. And I gave you a little quote in each of these that I won't read because you will. But within this, this is all about identifying the plants, understanding their distributions, understanding which grow together to form different habitats. And we do that through herbarium specimens, of which we have about 150,000 combined plant and lichen specimens in our herbarium. These are digitized and online, and more we even have scans of these. And these get over 10 million hits on our specimens each year, so that scientists around the world can learn about these plants. We also go farther to understand the relationships and the evolution of these different plants, because you know where they've come from, then you'll know better where they're going. And then understanding the threats and the changes to them. And here's an example of the value of these historical records in herbarium specimens. This is the California jewel flower. It's beautiful, it's federally endangered. And if you look at this map here in the little boxes, the blue ones are where it still occurs, and the red ones are where it's been extirpated. So having these historic records helps us trace this history and to work to conserve what's left and hopefully re-expand. So I'm going to give you just a couple examples of this kind of project, some studies in Santa Barbara County, and then some DNA barcoding and meta barcoding work. So Santa Barbara County is super rich floristically. We have just great differences across the county in things like topography and soil. And while a flora has been studied and a flora has been put together, we have a lot more work to do. And so our botanists are working on updating the flora of the Santa Barbara region. It will include keys and drawings and all of the things you need to understand what these different plants are in this county. And if you look at this map here and all these little green dots, this is where plants have been collected in this region. And when Matt, our lead botanist, created this map, he noticed right away that there are what he called botanical black holes, these areas that have not been collected before. And so we're aiming to fill those in and really improve our understanding of the flora in this region. So one opportunity to do that came in the Zaca and Jesucita fire scars. You can see here that the Zaca fire alone was almost 250,000 acres, and the Jesucita fire was right in the garden's backyard. In fact, it came through the garden. We did a number of things, including just basic biodiversity surveys, and we needed to get deep into the wilderness. This is why these were botanical black holes. And so it meant a lot of hiking, a lot of backpacking, but also going really old school with horses and mules to carry our gear and our big herbarium presses and all the things. So we got deep in there. It was beautiful. It was fun. It was an adventure. And we collected a ton of herbarium specimens, over 3,000 new specimens in those 2 years. And these, of course, are helping us to document and understand the flora of the Los Padres National Forest. And there in the lower photo, you can see these packed herbarium presses with all the different specimens that then go into our herbarium. So this other project is at an even finer level, at the molecular level, doing surveys also, but then using DNA barcoding to even deeper understand the flora and beyond of San Nicolas Island. So that starts by taking a single unknown sample and then running, extracting the DNA, running PCR to really amplify the DNA to a large amount, and then comparing those barcodes to a reference library in the Barcode of Life data system, so that everything gets much like you would see as like a SKU in the grocery store. It has a barcode so that even if it's difficult to identify, you can figure it out that way. And we had a big team of taxonomic experts heading out to San Nicolas Island, looking at the vascular plants, but also the bryophytes, the mosses and things, the lichenized fungi, and also the macrofungi, and then selected invertebrates as well. And so far we have 4 new bryophyte records, things that had not been recorded on the island before. Macrofungi have been very understudied on the islands. We uncovered about 113, possibly 2 new to science. And by we, I mean us and our partners. 25 new lichen records for the island. So even though here we are in the year 2022, there's lots to be discovered and things even still to be named. Okay, so goal 2 is to protect and recover California's most imperiled plants. And it might be things like this beautiful Santa Cruz Island bush mallow or this Hoffman's rock cress growing in our nursery. But it's important that we keep every piece because everything has a niche to fill and a job to do, and that tapestry of life can unravel really quickly. So we do that in a number of ways. First thing is just insurance policy. We take conservation seed collections of everything we can get our hands on, and as representative as possible of all kinds of different moms in different places at different times. And we get those into our freezer, and we could use those for research and reintroduction, and then again, just insurance. We do a lot of research and a lot of monitoring to really understand what are the threats and opportunities for these species. And then restoration and management. And just a couple of examples of projects. One is a really big project on the Channel Islands with a bunch of partners. And then one is focused on the salt marsh bird's beak. So this first one is working together to recover 14 different listed plants on the California Channel Islands. All of these wonderful different species that are found only on the islands and that are rare and endangered, and have a myriad of different challenges on the island, including introduced species like goats and pigs and sheep and deer and elk and whatnot. So being from your university, I'm sure you know about the Channel Islands, but just to make sure, these are the 8 Channel Islands off the coast of California. And this project covered 7 out of 8 of those. San Clemente had a lot of their own money to do this kind of work. And the work is really multifaceted, typically includes a lot of these different aspects for these different 14 species, including lots of surveys, just getting updated counts on these species and information about their habitats and trying to understand their trends. A lot of mapping and detailed mapping, and really kind of upping our game as far as the protocols we use to do that mapping. So one of our partners devised a wonderful system that's based on a grid so that the polygon you're searching is always consistent. Sometimes it involves investigating the genetics. Sometimes you have to sort out the taxonomy first, or look at population genetics to see if these small populations don't have enough genetic diversity. We always try to build in seed banking when we can, or for things that don't keep well by seed, we can add them to our living collection where we have representative individuals at the garden itself. And then of course the recovery involves propagating from either cuttings or from seed, and then restoration and reintroduction actions on the islands like seeding and outplanting. And this is a huge collaborative effort between not just the island owner managers, but also agencies and other nonprofits and for-profits. And we could not do it without all of our super dedicated volunteers at the garden as well. Just one little example of one of those 14 species. This is the island barberry, state and federally endangered, known only from Santa Cruz Island. It's presumed extirpated on 2 other islands. And at the time, only 9 known genetic individuals. Since, as part of this project, more have been discovered. But this involved emergency propagation of the island barberry, very dramatic, bringing helicopters in to get right to the spot we needed to. And then taking cuttings very carefully with all the metadata. And our crews collected nearly 200 cuttings to use in restoration and recovery efforts on the island, so we can start new populations that are genetically diverse. Okay, my other rare plant example is one of my favorite plants. That's the salt marsh bird's beak. And you can see it's beautiful. It's found in high salt marshes. It's a poster child for climate change and sea level rise, and it's been extirpated from 60% of its former populations. So those are highlighted in red on this map that shows where it used to be. And Carp Salt Marsh is one place that we've studied a fair bit, as well as Point Magoo. So for this plant, we've done a range of things, including conservation seed banking across the Central Coast Range, looking at pollinator studies and comparing to a study that was done in the 1980s, looking at the impacts of invasive plants, and looking at the distribution of seeds in the seed bank of the soil, and then doing some restoration study designs for the Navy. But the exciting, well, not in a happy way, some of the more interesting results is by repeating a study that was done in 1985, we found some evidence that bird's beak pollinators have declined. Of course, this is only 1 year of data, but so was the 1985 study. But in 2016, we observed only half of the different bee species that using this plant that were found in 1985, and only 5 individuals across, I forget how many hours of staring at these plots, looking for pollinators on this plant. So whereas Patricia Lincoln had found sweat bees and Sonoran bumblebees, we did not. We did find these 2 bees, the wool carder bee and the longhorn bee. And these big bees are really important to pollinate this plant. It does not reproduce without them. We next asked at Carpentry Salt Marsh, and I should mention that we did that study again for a second year, and it wasn't quite as bad, but still not good compared to Patricia Lincoln's study. Then we started a study at Carpentry Salt Marsh, which has an invasion of this European plant, European sea lavender. And it's really, as invaders do, it's displacing the salt marsh bird's beak at Carpentry Salt Marsh. And we were wondering, does it not only push them out physically, but is it also affecting the insect pollinators? So we set up some plots. And what we found, I think, is really interesting. So on the left, these 2 bees are native bees. And it's only the native flower visitors that we're using the bird's beak. Whereas on the right, in the 2 colors of gray, these are the insects that we're using, the European sea lavender. And these are both European species. So we have a European honeybee, which is itself invasive, and then we have a European flower fly. So it really split out almost 100%, 98% of the floral visitors to the sea lavender were European, and 100% of the floral visitors to the bird's beak were native species. So European sea lavender is supporting European flower visitors. So not only is it competing directly, but it's changing the pollinators. And if these European species are what the sea lavender is supporting, and it's not what the bird's beak needs, then it can go downhill kind of fast. So then we started a removal experiment at Carpentry Salt Marsh that is still happening, where we, in our plots, half of them we removed the sea lavender and half we left. And then we're repeating these methods and comparing in a before-after control impact study. So stay tuned on that. Okay, lastly, goal 3 is to conserve and restore Central Coast habitats and food webs. You know, it's just not enough to conserve land and set it aside. A lot of times we just have to do active habitat restoration to get, you know, the biodiversity back. So the idea here is to protect entire ecosystems and not just single species. So a couple examples of some projects in the restore category. San Nicolas Island invasive plant impacts. We did a bunch of prioritization to guide habitat restoration in some different fire scars. And then we're currently doing a restoration experiment in the Piru fire scar. So you probably already know this, but invasive species are one of the greatest threats to the diversity of life, mainly because they come here without their natural controls like insects and pathogens, and they often take over at the expense of the diversity of different species that support the food web. And invasive plant impacts then follow the food chain up. And on San Nicolas Island, this food chain ends at the top carnivore island foxes. And of course, I'm pulling on your heartstrings by showing you this cute fox photo because, you know, that's where it ends. San Nicolas has been hammered by humans, as have all of the islands, but it still hosts some really rare and unique plants and habitats. Things like this buckwheat that's only found on San Nicolas Island, lovely dune habitat that's shown in these pictures here. But crystalline ice plant, this Embryanthium crystallinum, has invaded the island, and it looks kind of like this with all the red. This is a South African species, makes the soil salty and just kind of makes life hard for everybody else. And here's where my plant-insect interactions background comes into play, is that insects and other arthropods are excellent indicators of invasive plant impacts and also habitat restoration success. And this is because they respond really quickly to change at really small spatial scales compared to other organisms. They also perform a whole variety of ecosystem functions, not just cute and fuzzy things like pollination, but also nutrient cycling, breaking down all that waste, also predation, which is also key to keeping things in check. And also herbivory, also important to keeping things in check, keeping one plant from taking over, for instance. Seed dispersal, and also parasitism. So predation and then some by laying your eggs inside other organisms and then eating them out from the inside. And our study showed that crystalline ice plant reduces plant diversity, and in turn, it reduces the bug abundance and diversity. And not just that, so it did this at 2 out of 3 sites, but what it did at all sites was either reduce the diversity or reduce the functional diversity. So the different kinds of jobs that are done by these different insects. And the patterns were really clear on which insects liked the crystalline ice plant, which tended to be really tiny little detritivorous things that would be really abundant, but not big and juicy like the food that the island fox needs. And so by looking at the diet of the island fox, it seems safe to say that by decreasing these larger insects, this probably is hurting island foxes. Okay, so second example, I want to revisit, you've seen this map before. Here's the Zaca fire, here's the Jesucita fire, and in that same project where we hiked around and rode mules and collected a bunch of plants, we had other goals as well. And that was to understand the invasive plants so that we could do some habitat restoration. And just a little diagram here, we aim for early eradication of invasive species. We want to catch them when they just got introduced. Because here's what happens, they get introduced and it takes a little while before they form a critical mass where we can actually detect them. So if we're lucky, we detect them pretty early and we can still eradicate these things. But it's usually not until over here, and this is time, of course, on the x-axis, where we actually start to notice that it's becoming a problem. And that's when we might begin interventions. But here is this zone, this middle zone, where eradication is possible, but really intensive. So we're putting a lot more time and energy and money into it. And then when you get to this part of the eradication curve, the cost is just too great and only local management is possible. So we really want to catch these things early on when their abundance is really low. But when you can't eradicate, because that often is not possible, we want to protect and restore. So what we did was we looked for invasive plant species and we hoped to catch them early, but we also looked for rare plant species so that we could prioritize when they were too far gone. So things like a different kind of jewel flower here. And after years of surveys, we found 237 rare plant locations. 604 weed locations. We made 8 conservation seed collections. We benefited from over 2,000 volunteer hours, over 307 people days. We did a whole lot of outreach. We made little videos, we posted images, we gave talks. So over 45,000 people reached. And in this process, we covered 735 miles. So if you mapped that on the state, it'd be from around San Diego to Mount Shasta. And we made lots of recommendations, such as, here's a map of the Zaka fire. And in this area, we find the only populations in the area of Medusa head, which is a bad invader. It looks like this. And we figured that it was early enough that it might be able to be nipped in the bud. And so since then, Channel Islands Restoration has done more investigation on this species. It co-occurred with a couple of rare plants like this jewel flower and this sunflower, a couple of things in the sunflower family, which made it all the more important to try to remove this invader before it got more out of hand. Since then, more recently, the Thomas fire burned as well as the Whittier fire, and we just finished a more recent project to survey that whole area. So we used a lot of volunteer time and community scientists to hike over 300 miles, searching much like before for invasive species, rare plants, and also erosion to prioritize habitat restoration. And working with the Conservation Biology Institute, using these data, they produced a model to prioritize habitat restoration actions across the fire scar. And so the next step will be to get funding to implement some of those things. And now, as of just like a month ago, we've been putting in plots, in this case, Josie Lesage, leading a bunch of staff and volunteers in creating these square plots, 16 paired plots, looking at controlling, restoration treatment versus a control treatment versus a reference treatment. And the idea being that with habitat restoration, can we benefit ecosystem services, things like soil functionality, invertebrate diversity, and carbon biomass storage after 1 and 2 years. So I mentioned that that kind of the icing on the cake is the education, the outreach, the mentorship, and the advocacy, and we do what we can there. We do classes, workshops, lectures, field trips. Several of us have been lecturers at UCSB. This is a field trip that we took to Santa Cruz Island for a Channel Islands class that I taught. Publications, presentations. We have an annual conservation symposium, which is coming up on March 5th. Topic is After the Fire, Habitat Restoration, and hope to see some of you there. This is one of our conservation awardees, Dr. Claire Kremen. We combine the symposium with presenting a conservation award to someone who is a conservation hero or heroine. We offer internships, we do mentoring. We now have a paid internship program for summer times, and websites, social media posts, committees, working groups, collaboratives, whatever we can do to get out there and put our knowledge to work to help make things better. And here's the poster for our conservation symposium coming up on a Saturday. Students get in free. And Dr. Carla D'Antonio is our keynote speaker and our conservation awardee. She's not only just a super scientist, but also just a really great mentor and really active in not only doing the science, but making sure that it's really applied, that you can really apply it in the real world to make a conservation difference. So I hope to see you there. And we also have lots of volunteer opportunities if you are ever interested. So I think that is all that I have. I'm going to move you guys over and I'm going to try to stop sharing if it will let me. Hi, Ciamara.
Hi, Denise. Nice to see you again.
You too. Hi, I see your background. Conservation Center.
Yes, yes, the nice pictures I took.
Thank you so much, Denise, for such a great talk. And it's a good thing to point out that we have Ciamaro, who's a biology major, who's here, was an intern who we supported to work at the garden. Actually, the research station supported, and working along with our campus and folks like Dr. Alvarado in biology, there's going to be even more opportunities and connections between our campus and the garden. So we're very excited about that. Thanks, Denise. I had one question, which was, you showed that great picture of your department and all the folks that are in it now and how it's growing so much. You couldn't even put on everyone's name. Obviously, you've brought people on in different positions and you've hired a bunch of people. And I'm curious your thoughts about in bringing on someone, particularly in like an entry-level position, someone who's, or even still an intern or kind of come out of undergrad in those positions, as someone who is a director and is making those decisions. If you had to pick like the top 3 things that are really important to you, kind of bringing someone on in that entry level in this field, what things might you prioritize?
Oh, that's a good question, Russ. Well, what 3 things would I prioritize? I mean, first, they have to have the right experience for the job. But also, if they're entry level, they might not have, they probably have some background, but they might not have all the job experience, right? So sort of the right person for the right job opportunity. And we're trying to really kind of build a ladder so that there's a range of opportunities. We can really mentor people just starting, but also, have opportunities for people with more experience. But probably equally important to me is that they're a good fit for the organization. It's really important. We ask a lot of questions about just like how you feel about biodiversity and why it's important to you, and what's your elevator speech for why conservation is important, and just making sure that this is someone who will fit in with the team. We all just really care about what we do. We're good people, and we want other good people to work with. So that would be number 2, I guess. Third really important thing. I don't know, Russ, but I'm going to share something, a tip of when we have interviews. My favorite interview question is, think about what makes you, you, and what are the characteristics that describe you? Like, give me some adjectives. And then think of an animal that has those characteristics. What are the characteristics and what is the animal? And I think more than one person has kind of laughed at that question or been terrified by that question. But I ask it because it really tells me something about that person in a deeper way. I just get a sense of what's your character, what's important to you, and in a way that might not work as well if I asked that directly, Does that give any insight?
Yeah, thank you very much. That's great.
Now I give out my secret, and now you won't be surprised when I ask that question.
If anybody else has questions, please jump in.
I do have a question. So as a director of conservation, I've worked under you, but I always wondered, what is the greatest, both the greatest challenge that you come across as director of conservation, and also, like, I guess the most satisfying part of the position that you have?
I guess I'll start with the satisfying part. I mean, just the brilliant, wonderful people that I work with, and all the good conservation work that we're doing. It makes me happy. We have Monday morning meetings where we all share what we've been up to and some highlights and things. And I really like that. I think it boosts me and everybody to hear all the good stuff that's happening. But I really like my job. I like the diversity of my job. I like strategic thinking and whatnot. And I like the science and I like the outreach. So it's all good. As far as challenges, I mean, one challenge is just Santa Barbara is so expensive, and paying people enough that they can live here is just really challenging. And the garden, we're a nonprofit. We're doing better probably than we ever have, but we're still kind of climbing out of a bit of a hole. And so we're doing our best to pay fairly and more each year, but it's really challenging to live in Santa Barbara. So I think that's a deep challenge.
I actually had a question too, if nobody else had something there. This was actually more of a curiosity. I know you mentioned the DNA barcoding that you guys do. Have you guys ever encountered anything that you haven't found a DNA barcode already in the system for?
That would be a better question for a conservation geneticist. I know that this work has been going on. They haven't finished that project yet. So I haven't necessarily seen a report that kind of sums that up. And I also haven't heard her say, oh, and we found, a new thing. That said, we do work with invertebrates on the islands a lot, and we've uncovered new things that, whole new genera that have not been described before. So I'm sure that we'll have some of that come about through the barcoding work, though it hasn't come to pass yet.
Thank you.
Yeah.
Another quick question that just came to mind. I've always wondered if someone was like looking, long-term, long-term career goals, at, I guess, a position similar to yours, what would they have to kind of keep in mind now, like that, while they're starting out, I guess, in their career?
Or like if they wanted this job?
Maybe not specifically your job, but like the position, the kind of the type of tasks that you do as a manager, as the leader of this whole conservation department.
I think what serves me well is my diversity of experiences. It helps that I got a glimpse into the environmental consulting world. It helps that I did so many different things on Catalina Island. So when we talk about understand, protect, restore, like I've done a little bit of that, in my career. So I think that diversity of experience helps a lot, because every day is just a little bit different. I might be working with budget spreadsheets, or I might be out sampling pollinators, or, giving a talk or something like that. So just, I guess, being as well-rounded as you can be. I know we all have strengths and weaknesses, but doing our best. Yeah.
Thank you.
Yeah.
I also had a quick question when it comes to invasive species. So I understand that they're bad for the ecosystem in multiple ways, but is there any way to reverse the effects, or can you only try to control the damage it does?
Yeah, good question. It really depends on the invasive species, because a lot of times they kind of change the game for the next thing. So like, for instance, that ice plant that I talked about, where it makes the soil more salty, it makes it harder for other things to colonize. And so you might need to kind of remediate that soil first, if that's the case in your system. On the other hand, you have other invasive species like feral animals on the Channel Islands, where the largest herbivore is a squirrel, native herbivore, right? And then we've introduced all these things: sheep, goats, pigs, elk, deer, bison, antelope, you name it. And it's just these plants are baby salad mix, because they lost a lot of their defenses to these mainland herbivores. And so the action there was pretty much as simple as get them off the island, and then things started to recover, except when for some of these rare plants, they were reduced down to such small populations that they couldn't make it, right? The populations weren't big enough to attract pollinators, or they weren't genetically diverse enough to produce seed or whatever. And so some of the work is just, well, let's grow more of them, and let's get more diverse populations and get bigger populations. Sometimes it's, not all that complicated, but then sometimes
thank you. Also, for the salt marsh bird's beak, and how you were talking about how the European sea lavender may be causing a decrease in the pollinators needed for the salt marsh bird's beak. If that's true, would you just try to remove the European sea lavenders, or how would you go about trying to resolve increasing the pollinators for the salt marsh?
Yeah, I'm glad you asked that, because some of the work that we did in the earlier study that I talked about at Point Magoo is we looked at not only which pollinators are using the bird's beak, but what other plants they're using. So if the bird's beak needs a specific big-bodied bee, like say a bumblebee, well, what else is the bumblebee using? So can we plant more of that? That's a little easier to get established than the bird's beak. And so we came up with recommendations like that. So things like a little a deerweed that it's easy to grow and supports the bumblebee really well. And so if we plant more of that while we're also trying to boost the population of the bird's beak, then we can hopefully provide more habitat for the bumblebee, which is also struggling. It can be complicated, but as far as that carp salt marsh, yeah, the idea would be to remove the sea lavender. And so that's what we're testing in those plots. It's actually what's happening with some partners like Tidal Influence and Channel Islands Restoration. They're removing the sea lavender, and I knew they were going to do that, and I took advantage of that to try to get an experiment in. And so we're testing that right now. Y'all have good questions.
Hi, I had a question. I'm sorry if there's any background noise. I am driving, but can you hear me okay?
Yeah.
Okay, great. I had a question about the use of pesticides like glyphosate and well, the like, but in your conservation and restoration areas, do you specifically, make any rules about whether or not that's allowed in the area, or do you find any connection between pesticide use and invasive species?
Yeah, good question. And we haven't studied that. Other people have studied that. So, and other people like the California Invasive Plant Council, they look at those studies and they come to a conclusion about, well, how strong are those studies? And do we feel like, certain herbicides are a justified tool? And so I look to them because they're spending a whole lot more time thinking about that. And they've concluded that, yeah, it's still an important tool. And so you can find more information on their website there. But really, as far as whether or not to use herbicides and pesticides, that's up to the land manager. So we follow, what they want to do. And a lot of times people will say, no, we don't want any pesticides. The problem is that it is an important tool in the toolbox, and it might be a little pulse of a negative when that just opens up a world of positive. And so like life is complicated, right? And you just have to weigh these different things and decide if the benefits outweigh the costs. And so it's really very much a case-by-case basis, but we do practice IPM, integrated pest management, and just use the least gnarly tool for the job that we can.
Got it. I think I may have phrased my question improperly, but I actually was referring more to the use of pesticides in general and how that creates an ecosystem depression as far as biodiversity.
Okay. Well, so if you look, for instance, at pollinator decline, which is a thing, around the globe, where more and more studies are showing that, and not just pollinators, but insects in general. You've probably heard of the insect apocalypse. So that's a thing. And there's just all kinds of reasons for that. And one of them is the indiscriminate use of pesticides. So I'm absolutely in favor of using whatever alternatives that we can. One thing that's different is, well, for instance, at our homes, we're some of the worst offenders. If we use pesticides, people think a little bit's good and a lot must be better. And we actually over apply way more than folks with training, like in agricultural systems.
Yeah.
And so, that's a problem. And so, yeah, I'm all for avoiding it if we can, but it's always just weighing the costs and benefits. And if we are using a little bit of herbicide, it's very, targeted application. We're not, putting so much on that it's going to run off or anything like that.
Certainly. Well, thank you for speaking with me about that.
Yeah.
I have a question. Hi, you've done so much in your career. Where do you see yourself going from now, the future, your program?
Thank you. Doesn't feel like it. It's such a big problem, environmental conservation that, there's only up to go. I don't know. I like my job a lot. I don't have any, immediate plans. I am really proud that we've grown and we're doing so much. so I just hope we keep doing that.
Awesome.
Denise, I had a quick question. You had said earlier in your slideshow that your first job does influence kind of the trajectory that your career can take. In what ways do you feel like it influences the trajectory that your career takes?
Yeah, well, it's you know, of course, things can go any number of ways, right? But if you're someone who's looking at an applicant and their CV, their resume, you know, you look to see if the experiences that they have had match the things that you need in the job. And so it kind of starts with that first job and that first thing on the resume, right? And then from there, it's easier to get the next thing and then the next thing. And so, you know, just as a general, it seemed like a general thing to say that, you know, so that first thing that you pick sort of matters. And a lot of times I know, unfortunately, it takes so much to make it these days. You know, hang in there, everybody. But you know, all the internships on the side and the volunteering and all the different things, like those are the places where you can choose more, maybe, because if you're volunteering, like, you know, you have more options in front of you. So you know, get experiences, figure out, you know, the best you can, that path, and then just kind of snowball that, I guess. Although, I mean, I took a winding path, so who am I to say?
Thank you. Does anybody else have any other questions for Denise before she leaves?
I guess I have a question. So speaking about first jobs, so my current job is I'm a paralegal, and I'm really interested in like biotech law and regulation and things like that, but just different things that encompass biology. But I've been having a hard time. I kind of have two loves, which is law and then biology. I want to be a field biologist, and I don't really know exactly how I can intersect them both. But since you know the field, I don't know maybe what your thoughts are on that.
Yeah, well, it's harder when you say the field biology, but I mean, there's so many great intersections with, you know, biological conservation and law, of course. I mean, a lot needed there for advocacy and just legal avenues, but also, yeah, you know, advocacy with folks in government, or gosh, there was something that came to mind earlier when you were speaking. It's harder to merge the field part. I feel like you could kind of do that on the side, you know, like anyone can do that on the side as a hobby, but like, I think that intersection is really important, and I hope that you find that sweet spot, you know. Yeah, I wish I could remember the other thing that came to mind, but.
Brigitte, you can go on the registration website, find my email, and email me. I know someone who worked for me as an intern, as a field biologist, when I ran a program on the Solomon Islands, who was a field biologist who wound up becoming, he went to Lewis and Clark, became an environmental lawyer, and now he's the head of the Center for Biological Diversity's DC office. Like, he works with Congress people and senators, like they're in the fights, like of the real struggles for environmental policy, and he tracked that path. He actually came, I had him come in and talk to the environmental law class a few years, you know, in my first year here, but I might be able to provide you with somebody who'd have a bunch of resources on that. So you can email me, or I'll just send you my email in the chat, and I'll connect you with Brett, because he might be able to address that question that you had.
Thank you so much, both of you.
It's such a sweet spot. Good, you know, go for it. You might not get in the field as much as you would like to, but it's such an important intersection. So, you know, people with those interests and the people skills, you know, that's exactly what I was talking about, was like, find your niche, the things, that intersection, the things that you're good at and that you love. y'all asked great questions, and all of you asked one, I think. So thank you very, very much.
Thank you, Denise, for taking time out of your day.
Absolutely.
We really appreciated you coming and speaking to us today. It was a great presentation.
Thanks. Right on. Well, it'll be on first YouTube presentation, I think. So that's terrifying.
You did really well, so.
Yeah. Excellent, excellent presentation. Yeah, I'll be up on our YouTube channel. And Jenna, can you remind folks when and who our next speaker is?
Yeah, so folks, on March 10th, we have Dr. Estelle Sandhaus. She is the Director for Conservation and Science at the Santa Barbara Zoo. So she will be speaking on the California condor nesting research and conservation efforts, which will be pretty exciting as well. So we're going to stick with that idea that we want to conserve as best as we can our California native populations. So we hope to see you guys there. Thank you so much for joining tonight, guys.
Thank you.
Thank you.
Thank you.
Thanks, everyone.
Hi, everybody.
Bye.
Hi, Dr. A.
Thank you, Allison. Did you bring your students? Did you rope a bunch of people in here?
She baited us with well, she baited the rest of them with extra credit. I came here for Denise. So.
Good questions, though. Good questions.
Yeah.
Really nice.
Yeah. Thank you for doing
Makes
that.
a huge difference when the speaker gets a lot of nice questions from the
Yeah.
students.
For sure. No, we really appreciate it. And we had great turnout. That's awesome. Yeah, I'm really, really, really happy for it.
Nice, nice.
Yeah, Allison, I don't think I missed anybody in the waiting room, hopefully.
Oh, yeah.
so.
No, I saw them coming in gradually. So I
Okay,
was keeping
good.
track. It's all good.
Yeah,
Awesome.
a couple of us had class until like 5:45, so it was like rushing to get to a computer.
Yeah, of course. Yeah. Awesome. All right, folks. Well, we will see you hopefully at 7 for Russ's Rub.
Yeah, I'm going on doing the thing for CIR. So yeah, thanks very much. And yeah, thanks for the recording, Genoa. Thank you, everybody.
All right.
And
Bye, everyone.
next time.
Yeah.
Bye.
Have a good night. Bye.